In this video, we are going to expand on the steps of DNA replication and focus on some additional information about the enzymes and the function of the enzymes. So just to recap, there are 9 steps in DNA replication. As you can see here in the diagram, I explained by using some color how these enzymes and other structures work together to allow the DNA strand to be copied on the leading side, the leading strand side, with continuous replication, and also on the lagging end, the lagging strand, so that you could have both of those sides of the DNA molecule copied. What I would like to do is to focus on the genes that code for these enzymes. So if we look here at helicase, it's important to know that helicase is coded by the DNA B gene. DNA B. Our RNA or primase, or RNA polymerase, that is, or primase, is coded by DNA G. Our DNA ligase is coded by the LIG or LIG gene. Topoisomerase or gyrase is coded for by an alpha subunit, first of all, of the gene GYR or GYR-A, and then also a beta subunit, and that would be B, so GYR-B. Our single-stranded proteins, our single-stranded stabilizing proteins that we drew in here on our diagram, as you can see over here. Of course, proteins, they will also be coded for by genes, and the gene involved here for the single-stranded proteins, single-stranded binding proteins, is the SSB gene. Next up, let's look at our pole 1, 2, and 3. Let me move this one down here for us, and just replace that one there. There we go. We can put our DNA pole 1 up here next to it. This enzyme is coded for, and sometimes also known as just pole A. DNA pole 2 is pole B, and then DNA pole 3 is much larger. It has 10 different subunits. So if we talk about our poles in sequential order here, DNA pole 1 fills in all the small segments during replication and repair. If we look at DNA pole 2 or pole B, it is an alternative pole to replicate damaged DNA. So it's not the main one, right? It's going to help wherever there's damage. So an alternative or alternate pole to replicate damaged DNA. Let's go next to DNA pole 3, the one we said has 10 different subunits. Normally, this is our main replication enzyme, and it is going to be responsible for copying the nucleotide base pairs. I do just want to point out that there are 9 DNA poles, 9 different DNA poles in eukaryotic organisms. So we cannot discuss all of that, of course, in this video. We're just going to focus on pole 3. But what is interesting about them, all of those pole 3s will add nucleotides in the same fashion. They'll add nucleotides to the growing strand in the 5' to 3' direction only. So what they're responsible here is for catalyzing the catalysis of those phosphodiester bonds in the backbone of the DNA molecule. All right, so DNA pole 3, let's look at the subunits here that is of note. We're not going to do all of them. I do want to point out that it has an alpha subunit. It has an epsilon subunit. It also has a theta. So that's with an E, theta subunit, and that's the sign for it. And these subunits form the core of the pole 3 molecule. The beta subunit, beta, of course, there's the beta sign or symbol, is a very important subunit. This subunit will be responsible for holding the DNA pole 3 very tightly onto the DNA strand. We don't want the pole 3 to become loose or, to separate from the DNA strand while it is copying. So that's important that it has this strong beta subunit. So that's why it's referred to as a beta clamp in our diagram. We said that was a beta clamp. So the clamp loader is a structure that will help to dislodge the primase once it is ready to be released, and then it will also help to recruit a beta clamp, a subunit from pole 3. So it's going to take off the primase and then make sure that the pole 3 is firmly attached with a beta clamp subunit. All right, now all 3 of these poles, pole 1, DNA pole 2, and 3, are what we refer to as exonucleases. So let's move down here a little bit where we have more space. All 3 DNA poles, right? We said, so this is 1, 2, and 3. They are exonucleases. Now, this is something we haven't discussed before. What is an exonuclease? Well, it removes nucleotides. That's important to remember. It removes nucleotides in the 3' to 5' direction. So if we just for a moment also define what is an exonuclease itself, maybe this will make more sense. It is an enzyme. Remember, we learned previously that if a name ends in a ASE, and in this case I wrote the plural, but a exonuclease, and we know that is probably an enzyme, right? That ASE that we see there refers to it being an enzyme. So many enzyme names end with ASE. So it's an enzyme that cleaves or removes nucleotides one at a time, and it does that, this is very important, at the end of a polynucleotide chain. Now let's contrast that with an endonuclease. Maybe you'll then see here what we are referring to if I do that. What is an endonuclease? If an exo removes at the end of a polynucleotide chain, then we can deduce from that an endonuclease will cleave those phosphodiester bonds in the middle, right? Not on the end, in the middle of our polynucleotide chain. Now, both these two types of enzymes, these two main groups are important to know about. We're going to also refer back to them in a later chapter when we discuss DNA repair mechanisms. Many of our restriction enzymes are endonucleases. So when we learn about EcoR1, for example, we'll mention that it is an endonuclease, a restriction enzyme. So all three DNA poles, remember, we said are exonucleases, and they have the ability to remove the nucleotides in the 3 to 5 prime direction. But there's something here we also have to add, and that is that DNA pole 1 also has 5 to 3 prime exonuclease activity. So that means it can remove the nucleotides in both directions, right? Pole 1 can do it in the 3 to 5 prime direction and in the 5 to 3 prime direction.